Hello everybody, we're going to continue on now with our story about what it takes to have a muscle fiber, a cell in one of your skeletal muscles, contract. And we've been learning about how there are neurons in your nervous system called motor neurons whose job it is to stimulate those muscle fibers to contract. So in the last video lecture we were learning about how, well, in order to make that happen, your motor neurons have to maintain a charge difference across their cell membranes, and we call that polarization, and the value for that is called the resting membrane potential for the motor neuron. So just to kind of review, we said the difference in charge across the membrane of a resting motor neuron is about minus 70 millivolts. And what that really means is in your intracellular fluids over here, here's your intracellular fluids, and here's your extracellular fluids on a motor neuron. If you total up all of the ions inside versus outside, you would find that overall there's more of a positive charge outside and more of a negative charge on the inside, and that can actually be measured as a voltage. Charge differences can be measured in as volts, and this is not a big difference. So the value is minus 70 millivolts. A millivolt is 1/1000th of a volt. Okay, so we talked about that on the last video lecture. We talked about how you have sodium leak channels and potassium leak channels, and then you've got these really important proteins called the sodium potassium pumps that govern that help you maintain that resting membrane potential. Super, super critical. You would not be able to survive if your neurons were not able to do that. Okay, and we also talked about how you've got a lot more sodium ions outside the cell versus inside the cell, and you tend to have a whole lot more potassium ions inside the cell versus outside, and maintaining those proper values in your ECF versus your ICF of motor neurons is super, super critical for your homeostasis, for your ability to be able to stay alive. We're also going to see that the same kind of thing happens in your muscle fibers. Your muscle fibers also have to maintain these differences in electrical charges. All right, so that's what we have under normal circumstances. What we need to do, what we need to have happen here is we need this motor neuron here that we're looking at. We need it to send a signal, an electrical signal, down here to these muscle fibers. So it's going to have to send that signal down its axon, that long appendage that it has. That axon forms branches, as we took a look at on an earlier video lecture. Those branches are reaching out and contacting and forming a neuromuscular junction with several different muscle fibers or muscle cells, as you see there in the diagram. And just as a review, all those muscle fibers are going to be in the same skeletal muscle. So if that motor neuron is in charge of controlling muscle fibers in the biceps brachii muscle, it will not be reaching out and contacting the triceps brachii muscle or any other muscle. So one motor neuron is only going to control muscle cells in one particular skeletal muscle. And so what is this signal that we're going to have to send down the axon? That is called an action potential or a nerve impulse. And what's going to happen here, an action potential means along a local area of the membrane, we're going to temporarily flip. We're going to go from minus 70 millivolts up to plus 30 millivolts. So temporarily, the ECF is going to become more negatively charged than the ICF. We're going to do a flip-flop. And then after that, we're going to have to reset that voltage so that we go back to a normal state. And then we're going to see that that temporary change has to move very rapidly from one end of the axon to another. And then once that charge difference, that action potential, reaches those neuromuscular junctions or NMJs that we have with our muscle fibers down there, that's going to result in the signaling that's going to tell those muscle fiber cells that they need to contract. So that's where we're headed here. All right, so what makes all of that happen? Well, we're going to have to do what's called depolarization of our motor neuron membrane. And all depolarization refers to, so we say that the cell membrane of a motor neuron or a muscle fiber is polarized when you've got that charge difference. You've got more negative charges inside, more positive charges outside. That's said to be polarized. Depolarization is what we were just talking about, where you start to lose that charge difference. That's called depolarization. So instead of the charge difference being around -70, it starts to go up. And that happens locally. I'm going to change my color over here for a second, maybe over to red. So this motor neuron that we see here, in Unit 5, we're going to learn about how neurons receive signals from other neurons. But you guys know your brain is ultimately the part of your body that's going to have to tell these motor neurons that are living in your brainstem or your spinal cord, hey, you guys need to send a signal over to the muscle fiber so that they contract. So the motor neuron itself is going to be receiving signals from the axons of other neurons. I'll kind of draw those in there. And when we say that it's receiving signals, what's happening is you're getting some local depolarization, like right around those contact points where that motor neuron is receiving signals from other sources. And so what does this mean? What's happening there? When depolarization occurs, you've got sodium channels that open up as a result of some sort of stimulus. Okay, so they open up a little bit. So we'll do a little erasing there to show that sodium channel opening up. And then what happens, you got sodium ions out here are going to start flowing inside the cell. Okay, so this is a sodium channel in this case, and it opens up as a result of some stimulus, and it only lets sodium ions through the channel. It doesn't let anything else come through. And as we recall, we've got more sodium out here than in here. So which way will the sodium ions diffuse? If that channel opens up, they're headed in this direction. They're going to head inside. All right, so what's going on here? All right, so we said we've got a greater negative overall charge inside versus outside, and that value is, let me erase this for a second over here, and we said that value at rest, when a motor neuron is not being stimulated, it's around -70. Well, if you start letting sodium ions flow inside, what's going to happen to all of your overall charge across that membrane? You're letting more positives inside, so the inside is going to become less negative. Okay, so if you were taking a look and numerically measuring the voltage in these areas over here where the motor neuron is being stimulated, you would start to see that value increasing, and that increase is called, this is depolarization. Okay, so we're starting to depolarize, lose some of that negative voltage that we have across the membrane in these local areas. Okay, well, these are ions, so these sodium ions that are coming inside the neuron, well, guess what? Ions can flow in currents. These ions are attracted, they're positively charged, so they're attracted to all these other negatively charged ions that are sitting around in your intracellular fluids. So even though they're flowing in over here and over here and over here, they're going to start spreading. If you keep stimulating this neuron over and over and over again, they're going to start spreading throughout the intracellular fluids of the motor neuron. And here's where we need to get them to go. We need them to go right here. If you guys remember, that is called the axon hillock. That's where we need them to wind up, because the axon hillock is going to be the decider about whether or not we have an action potential, a very rapid flip in the charge across the membrane that's going to zap all the way from one end of the axon down to the other. That's what we're trying to get to. All right, so let's move on over here to our next slide. Well, here's a little summary here. Okay, so we're having some changes in the membrane potential. Again, that's just a fancy way of talking about that voltage. And so we said, okay, this motor neuron is receiving signals. It's being stimulated by other neurons, receiving communications from the brain, and that communication causes certain types of sodium channels to open up. They start letting sodium ions inside, and so our voltage increases towards zero. So the inside of the membrane is becoming less negative than it normally would be at your resting potential. And again, it's due to movement of sodium ions across the membrane, usually. As this keeps happening, if it keeps happening, it's going to increase the likelihood that we're going to have an action potential. And now our motor neuron can send a signal to the muscle fibers. All right, so again, we need this to happen. We need this depolarization to reach the axon hillock. So it's got this diagram doesn't really have a good axon hillock in it. Well, it does. You can see the little wider area there where the axon is attached to the cell body of the motor neuron. All right, so what's going to happen if the axon hillock depolarizes enough, and we'll see how this works, an action potential is going to get started. And again, the action potential means we're going to have a very rapid flip-flop of the membrane voltage in a local area from -70 all the way up to +30, and then that is going to spread all the way down the axon towards muscle fibers. Some other key things about an action potential. It starts here. It very rapidly spreads all the way to the end. It does not decrease in magnitude over distance. Okay, so you're going to have a flip from -70 to +30 all the way from one end to the other. And also, this is how you have communication within the nervous system. So motor neurons communicating to skeletal muscle fibers, and when we get into Unit 5, you guys are going to see this is also how neurons communicate and send signals with each other. You've got lots and lots and lots and lots of networking that goes on between your neurons. All right, let's move on to the next one. All right, so returning to our main story over here. So we said this motor neuron is receiving signals from others, and that's causing some depolarization to occur. Membrane voltage is increasing. You got sodium ions flowing inside the motor neuron, and if enough of them get in there, they're going to reach the axon hillock. They're going to flow through those intracellular fluids. They're going to diffuse through there and reach the axon hillock. All right, and then at our axon hillock, what we've got some special types of sodium channels called voltage gated sodium channels. We have those at our axon hillock. Okay, so let's say, let me kind of flip colors over here again for a second. We've got all these sodium ions that have been diffusing through the inside of the motor neuron here because it's being stimulated and those sodiums were let in. So if they reach the axon hillock over here, what starts happening to the membrane voltage here in the axon hillock? It started out at -70, but now it's starting to go up. The more sodium ions that reach the axon hillock on the inside, the higher and higher and higher your membrane voltage is going to go. All right, so these voltage gated sodium channels, what they do, they're actually, as the name suggests, they're sensitive to voltage. When you reach about minus 55 millivolts, these sodium channels flip open. They're gated. So they flip open when you hit about minus 55. And what's that going to do? They're sodium channels. So guess what they're going to do? They're going to rapidly let a whole lot more sodium ions into the motor neuron, right around the axon hillock. Those are going to rush in. And there are lots of them sitting in the axon hillock. So you have a whole lot of sodium ions that are rushing in really fast. So what's that going to do to the membrane potential right around here at the axon hillock? It's going to very rapidly increase like that, really quickly, right around the axon hillock in this local area. We're going to see that happen. Okay, and this is our action potential that's happening. We have now completely flip-flopped. We've gone from minus 70, we kind of maybe slowly went up to minus 55, but once we hit minus 55, these voltage-gated sodium channels swing open, and there are a whole lot of them there in that area, and all these sodium ions come rushing in, and now our membrane potential flip-flops. We go very quickly up to plus 30. So inside the cell, you're now more positive on the inside than you are on the outside temporarily. That's our action potential. When we get up to plus 30, we can call this complete depolarization. I won't write out depolarization entirely, but you can see what I mean. All right, so I erased my drawing here, so we want to return to the story for a second. Let's come back. So what we've had happen here at the axon hillock is right around the membrane there, we have completely depolarized our cell membrane. So we now have a difference in charge across the membrane of plus 30, and that means the inside temporarily is a little bit more positive than the outside. All right, and we have those voltage-gated sodium channels that we were talking about. That's a sodium channel. And that has been allowing sodium ions to rush from out here to the inside of the cell. Now when you hit plus 30, what happens is the sodium channel gates close. So the sodium channel closes back up. All right, so that prevents any more sodium from rushing into the cell from outside. Now we need to reset our membrane here around the axon hillock back to the normal values, because motor neurons often need to send signals over and over and over and over and over again, or it may need to rest for a while and send a signal later. So we've got to reset our membrane back to normal. We've got to stop this complete depolarization that's happened. Okay, so also in the membrane here, what we have are voltage-gated potassium channels. I'll draw one of those in green. Okay, that's going to be a potassium channel, and it's voltage gated. And what happens is they open up. Guess what? When you hit plus 30, these potassium channels open up, and you've got more potassium ions inside the cell versus outside. So which way are they going to flow? They're going to flow out. They're going to head outside from the ICF to the ECF. So what's going to happen to the voltage, the charge difference across the membrane? We're letting positive charges out. Okay, now right now the inside in this local area is more positive than the outside. So our charge difference is going to go back down. Notice that when I drew that in, I actually went below minus 70. And that's actually important for action potentials as well. This process of returning back to near normal is called repolarization. I won't write out the Asian part. We're repolarizing the local area of the membrane by opening up those potassium channels. Now when you get down here, these potassium channels are going to do just what the sodium channels did. They're going to reclose up. And then we're not going to let any more of those potassium ions out. That prevents us from just letting tons and tons and tons and tons and tons of potassium out and totally screwing up our the charge difference that we have there across the membrane. Okay, I'm going to go ahead and move over to the next slide. This is giving you another look at this. There's a link there to an animation. I'll put that in Blackboard as well, which will walk you through this story. And again, a lot of times it just helps to see it animated. So this is a graph showing you the changes in the charge across the membrane. So again, we started at -70. At the axon hillock, when we had enough of those sodium ions flowing in from other parts of the motor neuron, enough that the local voltage reached -55, that's called threshold. That's our threshold voltage. That's the voltage at which those voltage-gated sodium channels swing open, and then boom, you let so much sodium in, you rapidly depolarize the cell membrane, and you go up to about +30. Those voltage-gated sodium channels shut at +30. They won't stay open anymore. But then your voltage-gated potassium channels open. You start letting potassium ions out. And that allows us to return close to normal. But if you notice, we dip below -70. So for a temporary period of time, the cell membrane is said to be hyperpolarized, overly polarized in that area. And when that happens, when you're hyperpolarized, it's more difficult to generate another action potential. Temporarily, you're not going to be able to do this action potential event if you're hyperpolarized. It would be more difficult for you to reach this threshold value over here that you need to reach if you're going to swing open those sodium channels and let all that sodium come rushing in. But we can't stay hyperpolarized. We've got to return our membrane back to its normal resting state. So you see there's kind of a gradual increase. Now these processes are taking milliseconds. MS means millisecond. That's thousandths of a second. So all this is happening very, very rapidly. During this period, what's going on is your sodium-potassium pumps are running. Okay, and they pump sodium out, potassium in, and so you go back to your normal state across the cell membrane where you have greater amounts of sodium ions outside, greater amounts of potassium ions inside, and that brings you back to -70. And now if we need to, we're ready to have another action potential here at this local area. Okay, so this, I believe this slide just gives you kind of a text summary of what I just said. And again, you've got to, in order to have that action potential where you flip up all the way up to +30 millivolts, you've got to reach that threshold value. Okay, so you have different kinds of sodium channels in the motor neuron membrane. They're going to show you those on the animations and talk about those in the textbook more. We're not going to go quite into as much detail as you guys have in your textbook. But there are different kinds of sodium channels. For example, when a motor neuron is being stimulated by another neuron or something else, that opens certain kinds of sodium channels that let sodium in. In order for us to do an action potential, though, we've got to open those voltage-gated sodium channels right around that axon hillock. Those have all got to swing open to let all of that sodium, all of those sodium ions rush in and allow us to go up to that +30 millivoltage that we need for the action potential. All right, and then that slide is just summarizing for you what I talked about with repolarization. And as I mentioned, you do become the local area of the membrane there is going to become hyperpolarized temporarily, where you go below -70. Now, there is another good thing about that. Temporarily, you want it to be more difficult to generate an action potential. You don't want an action potential to just continue on. You don't want it to be super easy to generate another one. It also prevents backwards flow of the action potential, because we're going to see we want our action potential to flow this way down the axon. If this is our axon hillock over here, we're going to want this signal to travel from one place to the next, to the next, to the next, down the axon. We don't want it going back this way. So by hyperpolarizing, it makes it more difficult to do another action potential at that particular location. So it helps out with that as well. All right. And I think I have covered all of this information pretty well for you. I know this is one of the more difficult topics you have in anatomy and physiology, so you just work with it and watch your animations. If you're reading along in the textbook, just be forewarned, they're going to go into a little more detail than what you actually need. But here's a summary about what the sodium-potassium pumps are doing. And I think I pointed this out to you on the last video lecture. Those sodium-potassium pumps, they have to use ATP to do all that pumping. This is where a lot of your ATP winds up going in your nervous system. Much of the ATP our body makes is devoted to running those pumps, believe it or not, so our nervous system can function properly. Okay, so I know action potential is kind of a heavy-duty topic, so I'm going to stop this particular video lecture there and let you review. And then in the next video lecture, we're going to talk about action potential propagation, another term for spread. How does this started at the axon hillock? Well, how is it going to travel down the axon of the motor neuron? So that'll be the subject of our next video lecture.